Good afternoon, everyone. Thanks, Mary, for the introduction. This is really exciting. Thanks to Nanopore for the opportunity since I did my PhD here. And I have the chance to share a couple of updates on a recent work that we are doing on investigating structural variations and complex cancer genome using Oxford Nanopore sequencing. So cancer is the second leading cause of death globally and was responsible for an estimated 9.6 million deaths in 2018. Cancer genome harbor genome instability, leading to widespread copy number alterations, structural variations, and chromosomal fusions. Complex rearrangement and catastrophic events such as chromothripsis, breakage fusion breach are quite frequent in cancer genome. So structural variants are variants larger than 50 base pairs. So the primary classes are like, deletions, duplications, inversions, insertions, and translocations. But in cancer, it could be much more complicated. So there are much more complicated classes, which in general we call them complex SVs. So they could be clusters of breakpoints enriched within specific regions of a chromosome, or sometimes multiple chromosomes are involved. So for the past couple of years, I've been studying complex regions of the genome in cancer. So, we previously we were really focused on the short read sequencing rather than long reads. So what we have been done using, like for the short read sequencing, we usually study different levels of the data to study complex regions of the genome. So for that, we were using copy number alterations, B allele frequencies, and structural variant data. So this is an example of the circus plots of the esophageal cancer sample, which we recently published. So if you consider the 12 o'clock, from the right side, you can see the chromosome 1, 2, 3, 2, X and Y. And the second layer is like copy number alteration, which is the red is loss and the green is gain. And then is B allele frequency, and in the middle are the structural variations, which is color-coded based on their type. So you can see like the chromosome 8 is very complex. There is a very, like the cluster of SVs on that region. By looking at the CNV, B allele frequency, and the cluster of SVs and the type of SVs, this is one of the examples that it was really hard to understand what's going on. So the thing is, short read sequencing is limited. So they are limited because they are just a few hundred base pairs. They have limitation in resolving in SVs while they're in the repetitive regions, and also there are limitations in phasing information. So with long read sequencing, we expect because we have like the longer reads, it covers the actually longer range of the region, which is like the structural variants are located there. And therefore, we expect that we have potentially much simpler analysis and more accurate genome assembly. So it's kind of like more picture of the jigsaw puzzle. So what we are interested in is like detecting SVs with higher resolution and improving the classification of complex SVs. So what we came up with, it was an in-house like approach or pipeline, which is consensus somatic SV calling using Nanopore. So we know that there is a lot of SV callers. Some of those are much better than the others, but they all have their pros and cons. So what we have done, we took sniffles 2, QSV, and SPIM. We definitely did the first filtration steps. And we have done the joint or consensus SV calling. So this is a bit tricky, challenging in long reads, because it's not as simple as what you do in short read sequencing, like you can use just BCF tools and do the consensus. So we did a lot of benchmarking, and after that, there's a lot of filtration steps, and we have like a consensus SV calls. So after that step, we have the somatic variant calling, which you have the tumor normal consensus SV calls. We just have called the somatic over the disease germline or normal variants. There is an optional step which if you have the matched short-read sequencing, you can see whether the breakpoints had any evidence, or you can see the evidence in whether it is available in the short-read sequencing or it's not. But this is the optional step, and there is the annotation step, and you finally have the consensus annotated SP calls. So we use these final consensus SP calls, and we actually use a couple of criteria. The time is really short in this talk, so I can't really go through that, including inter-distance breakpoints, size of the SPs and SP types, to classify or to identify the classifications of the complex SPs and the complex regions. So I wanted to just show a couple of examples that how this actually improved to some extent, our classification. So this is the case one, which is, I'm going to show you two different cases. This is in collaboration with the Zero Childhood Cancer Project. So the first is neuroblastoma, which is a cancer type which happens in the nerve cells around the adrenal glands. So the circus plots, if you consider 12 o'clock, so the chromosome 1 is clockwise is 1, 2, 3. And so we have shown the outer layer is the inter-breakpoint distance. Then we have the density plots of the duplications, deletions, and insertions. And in the middle, we have the links of the intra and interchromosomal SVs and also inversions. And the reason actually we are separating them, because in terms of the catastrophic events in the cancer genome, the ways of the different SV types are quite different. So inversion and translocation are quite important. And the labels, as you see, the blue are those which has the novel breakpoints. We didn't have the evidence in the short-read sequencing. The red ones are those which we saw the evidence of the breakpoint, but unfortunately we couldn't classify based on the short-read sequencing, and the black is like we previously detected. So as you could see, there is like a very complex region in the chromosome 2, and we could identify inverted duplicated deletions in the regions of the ERBB4 gene, MAP2 and IDH1. So what is the effect? These inverted duplication deletions are very much involved sometimes in the catastrophic events, and they could disrupt tumor suppressor genes, activate oncogenes, and also they can create a cascade of secondary SVs. This is the second case, anaplastic ganglioma, which is the very rare brain tumor. So this is another example, so the codes are the same. So as you could see, there is a very, very complex chromosome 9 and 10. So interestingly, we could like identify inverted duplication in chromosome 9, which the GATA3, which is an important driver genes in cancer, it was affected. Also, we identified an inverted translocation between chromosome 3 and chromosome 9 and 3, which was leading to a fusion gene of NTRK2 milk, which NTRK2 is quite an important gene in cancer. So in terms of chromosome 10, we identify many inverted duplicated deletions, but I think because there's a lot of genes involved, but it's not from the same species, so because it's like kind of multiple links, the exact final fusions, or the final picture, we really need to check the maybe de novo assembly or fusion genes in the RNA level. So for these purposes, we actually try to do the de novo assembly. It's notable that de novo assembly of cancer genome is very challenging. It's not similar to the, I think, human normal genome. The good thing for us, it was for the first time that we could reach and we actually performed that with Shasta, and when we mapped back to the HG38, we could reach around the 84% of the coverage. But we still, because if you see the big components, and it's actually chromosome 10, which was really complex, it was there, and there was very interesting that we could see a lot of connections and understand which species are finally what would be the graph and what's the connections. But unfortunately, we are still dealing with this is a diploid genome. So we are still working on whether it makes sense or not. So we are adding phasing information, but this is actually ongoing work, so we don't know how much that could be improved. So in summary, we established a consensus somatic SV calling workflow to identify and classify complex SVs. I showed you a couple of examples. We believe that genome assembly could enable us to gain more picture of the jigsaw puzzle of the chromosome and complex rearrangement, but this is actually very challenging, and we are not really sure how much we could success in that. And this is really ongoing work, and we actually have to improve all of our analysis in future. And also we are doing on more samples and more cancer types, and even deeper, and also using super accuracy at this moment. We previously didn't have super accuracy of the copy, so yeah. I have to acknowledge all the people that were involved in Zero Childhood Cancer Project, sponsors, and my team and group leader, Ara Devsons. We are from the Genomic Technologies group of the Galvin Institute. Thank you.